I hope you all are well. You can see different file formats in here. So look at, this is FASTA file format. This is FASTQ file format. This one is our Genbank file format. It contains first header information, then the feature information, then our sequence. And this one is the EMBL file format. So here is the sequence, and this one is our CLUSTAL file format, that is actually contains the output of multiple sequence alignment. So these all are different file formats, but we can read these all files using only a single function. So that is actually our bio.seqio module. That is from biopython we can use the seqio to read all these files. So not only these files, so here you can see different file formats that we can read with the seqio module. So mainly we will focus on CLUSTAL, EMBL, FASTA, FASTQ and Genbank file today. So first we will import the seqio. Then first we will read our FASTA file. So our FASTA file was sequence.fasta in the data directory. To read we will use the seqio.parse. Look at this carefully. We will talk about seqio.read later. So here, in FASTA format we have the sequence ID, this one, and other things, that is this portion is actually description, and this is our sequence. So we can read them as like record.id, then record.seq, this one, and then record.description. So how we can get this information. After this we can read our FASTQ file. In FASTQ file we have a identifier at first, then we have the sequence, and then our quality information. So these are quality information. So record.id will give the identifier, then record.seq will give the sequence, and we can get the quality information by record.later annotations. And for the Genbank file, here we have header information, then these are features. We have features as source, then gene and ACDS. We have a few information here. This is the trans protein of these CDS, and here is the complete sequence. For that, record.id will give us the accession ID, then record.seq will give us the sequence, this one, then the annotation, that is our header actually. So we can get the annotation using the record.annotations. Then our features, so we can access all the features it has. That is, our first feature is type.source, then our feature was gene, and the corresponding information of that feature. So here you can get the gene name and the CDS. So here is the protein sequence and the EMBL sequence. So this actually contains this is the ID, this is the linear type and RNA molecule, and here we can get information, this how many BP, that base bases are in the sequences, and in the bases how many adenine, how many cytosine, how many guanine, and how many thymine, and if any other later. Okay, so here are actually total 69 bases. Recorder ID will give us the ID, then the description, then our sequence, this one, and annotation, that is the corresponding information, and our feature, actually what features it had, how we can read the EMBL file. And if we look at the CLUSTAL file, so here we have 4 sequences, and for 4 sequence we can see the match and mismatches here. So these actually indicate the match, and these one mismatch, that is here we can get 3A, but a mismatch is T in this position. So we can read the CLUSTAL file also, so like this. So this is our first sequence, and this is the ID, second sequence and ID, and thus fourth sequence and ID. So look at here, we just used seqio.parse in every sequences, in every read. So if we are sure, that is if we are sure that our file contains only one sequence information, then we can use the read, sequence dot read. And if it contains many, that means, so if I show you here, here is the okay, so here is the another FASTA file that contains 5 sequences. So to read these we must use the seqio.parse, but for this we can use the seqio.read. So if we consider the Genbank file here, it contains only one record, so we can read that using the seqio.read function also. So just let's read that. So as it contains only a sequence, a record, we just need to change here. We are reading the Genbank file using the seqio.read, and now we will print the other information. So this is the difference actually. So if there are only one record, we can use that using the we can read that using the seqio.read function. Otherwise we need to use seqio.parse. Thank you for watching the videos. Happy learning.